All right, here we go again. So, just to review, we talked about imine formation last hour. We now know how to take an aldehyde or a ketone and an amino group and get an imine. With PLP, you can convert by tautomerization or 2 proton transfer steps the aldimine from the PLP aldehyde to the ketamine, which you get by deprotonating the alpha carbon and then reprotonating on the other side of that alpha carbon, either directly as shown here or via a resonance stabilized intermediate shown here. Here is the ketamine. Now, here's the jump. What we're going to do is hydrolyze that imine. So you've done imine formation. What we want to do now for, I think, question 3 of your quiz is figure out how to hydrolyze the ketamine. You're going to use water, and your steps here are going to be exactly the reverse of the steps to make an imine. That is, you're going to break apart the nitrogen and the carbonyl carbon, breaking both sigma and pi bonds. You'll break apart the oxygens on H3O and hydrogens on H3O plus. Then you'll put the oxygen from H3O plus in place of the nitrogen in the ketamine, and then you'll put the protons from H3O plus on the nitrogen. So I want you to go ahead, based on what I told you, and running the click and unclick things backwards, predict the products of this imine hydrolysis. You're going to get 2 products. One is going to come from PLP, but it's not going to be PLP anymore. And the other is going to come from your amino acid, but it won't be the amino acid anymore. See what you can come up with. And just for completeness, let me bring our imine formation rules. We'll paste them here, enlarge them, only now we're going to call them imine hydrolysis rules. We'll start here. Unclick, unclick, click, click. And I need to swap out the breaks and the makes. Sorry, I decided we were going to do that. Okay, anybody need a hand? Anybody got a question? No, it's got to be different from the starting materials. Yeah. No. This nitrogen, the oxygen H3O plus, pull up all the hydrogens, put the oxygen in place of the nitrogen on the top, then put all the hydrogens on the nitrogen. Got it? On one of those vending machines, you have to swipe your card like 60 times before it takes. Hmm? Oh, fun fact, it really works better if you swipe from the bottom up. Okay, you guys have an answer? Anybody wish to draw an answer? Okay. Yes? You have no idea? I called your bluff. That's okay, getting stuff wrong is the way we learn. I'll tell you a story about getting stuff wrong in a minute.
I'm using H3O, right?
Yep.
Okay. Wait, I don't have to just show the arrows?
No arrows, just products.
Just products, okay.
This is the reverse of imine formation.
We're gonna go with, oh goodness, okay. I'm just gonna draw.
So close, we want the oxygen to be in place of where the nitrogen is. Yeah.
Then,
And go ahead and get rid of those hydrogens, because those hydrogens are actually going to go on to the nitrogen. And if you want, you could use an R for the rest of ELP. Good.
I don't know where the extra hydrogen went, but
You could put it on the amine as R and H3 plus.
so.
Okay, great. Thank you. So do you see the symmetry of this reaction? You started with an aldehyde and an amino acid. You brought them together to form an imine. Then you tautomerized to get the other imine. Then you broke those apart, but now it's the other bond that breaks. Do you have a question? Yeah.
Yeah, do you mind just like running through this a little bit?
Yeah.
I'm just stuck on like where the additional oxygen came from.
Okay. Yeah. I'm erasing this so that I can draw the structures as we're going through, but the answer's right. So that eraser thing is going to drive me absolutely insane. Dang it. Well, somewhere we'll figure it out, but. Okay. So, when you hydrolyze an imine, remember, it's the bond, when you hydrolyze an imine, it's the bond between the pi bond and the sigma bond between the nitrogen and the carbonyl carbon that you're going to break. Okay? Conceptually, what I will do is I'm going to unclick the carbonyl carbon from the nitrogen, put it there. In its place, In its place is going to go an oxygen. That's the oxygen from water, or H3O plus. And then over here, I've got the rest of the molecule. Only now the nitrogen is not doubly bonded to anything. Instead, it's bonded to the 3 hydrogens from water. Yep. So do you see how this is exactly backwards? Do you see that in our starting materials? And what I will do actually, let's put all of this on the same screen so we can review it easily. That's hydrolyze the imine. Then we had a make the imine. Oh, shoot. Well, so first we make the imine. And the way we make the imine is using the PLP aldehyde. All right, so we make the imine from the amino acid and PLP. That gives us the aldimine product. And then, of course, once we do get to the aldimine product, we can do the tautomerization step, which gives us, instead of the aldimine, what we've called the ketimine. And once we get to that ketimine, we can hydrolyze it to get a new amine and a new ketone. Yeah?
So I guess I'm still a little confused why we hydrolyze the double bond. Because I feel like whenever we've hydrolyzed before, it's always been like just a single bond. So I'm just...
Yes. So when you've seen hydrolysis before of an ester, you've broken a single bond. The reason that worked is because you had a nucleophile attack a carbonyl carbon and there was a leaving group, and the leaving group was the thing that was single bonded to it. Imine chemistry is a little bit different, and to understand it, you need to understand the mechanism. But basically at this stage, you're going to have an intermediate after water attacks. And I'm skipping several steps ahead, so I'm sorry. I'm just going to show you the relevant intermediate. R is for the rest of PLP. You've got an intermediate like this. And what will basically happen from this intermediate is a base from the enzyme will remove the proton. Electrons will kick down. The nitrogen will leave. And on its way out, it picks up a proton from an acid group. That's not all of the mechanism, that's part of it, but that shows you why it's the double bond that's broken. The pi bond breaks in the addition step, the sigma bond breaks in the elimination step. Yeah?
So of all the things that carbon is bonded to, the amine's the best leaving group?
Of all the things the carbon is bonded to, is the amine the best leaving group? Yes, in part because it can leave as the protonated ammonium ion, which is pretty stable. All right, so notice the symmetry. the aldehyde used to be on PLP, the NH3 used to be on the amino acid. Made the imine, tautomerized to the other imine, hydrolyzed the imine. Now look what's changed. The amine is on PLP, and the carbonyl is now on the amino acid. And that's why this is called an aminotransferase reaction, because the amino group, which used to be here on the amino acid, is now here, not on the amino acid. So this the product is no longer PLP, it's called pyridoxamine. It's the amine version of PLP, and this is a nitrogen carrier. And over here, alanine has become, this is something that you've seen before, it's pyruvate. This is an alpha keto carboxylate. And that can go into the pyruvate dehydrogenase complex and the citric acid cycle. And you can metabolize it like that. So one of the questions in your problem set is going to ask you, how much ATP do you get from metabolism of the amino acid alanine? You got to do the aminotransferase reaction first. That doesn't require any ATP. That converts alanine into pyruvate. And then pyruvate can go to be broken down via the pyruvate dehydrogenase complex and the citric acid cycle, or it can be used to make glucose in gluconeogenesis, which we didn't talk about. My son, the missionary, knows about gluconeogenesis because he took a bio 100 level class, and because he's a weightlifter and he's worried about when you fast, your body starts to do gluconeogenesis and turn protein or muscle into glucose, and that's a bummer if you're a weightlifter. So anyway. Okay, I want to make sure you've got the big picture. The keys are here. First, make the imine with PLP and the amino acid. Tautomerize from the aldimine to the ketimine. Switch sides, switch which side is the imine just by proton transfer. Then hydrolyze the ketimine. That takes the amino group that used to be on the amino acid, and now it's over here with PLP as pyridoxamine. And that takes what used to be your amino acid, alpha amino acid, and turns it into an alpha keto acid. Okay, questions about that? You're going to see that it's these alpha keto acids that are useful in metabolism. So you can either get energy directly from the amino acid after it becomes the alpha keto acid, or you can convert it through gluconeogenesis, which we're not going to talk about, into glucose, which you can then use in the usual ways. All right, let's see how well you did. I want you to think through the whole process now. Well, you've thought through part of the process with aspartate. Aspartate is the following amino acid. You've actually already, at least on the front page of your quiz, you've already drawn the aldimine that you get from aspartate. So I'm not necessarily worried about that. I want you to think through that process. I want you to do imine formation. Draw the aldimine, which you've already done probably, so check that off. What you maybe haven't done for aspartate is tautomerize. So using what we've talked about before, I want you to tautomerize to the ketimine. And then I want you to take that ketimine and hydrolyze. And you should get out an alpha keto acid, and you should also get out pyridoxamine. When you get out that alpha keto acid, it should look familiar to you. I want you to see if you can recognize it as something you've seen before from a molecule in the citric acid cycle. Okay, so the aldimine should be on the front page of your quiz. I can go get it. It's here. Only I R grouped the PLP, and you're welcome to as well. I'm not asking for arrows. I just want to see products. So show me first the tautomerized ketamine. And then I want to see the products, pyridoxamine and the alpha keto acid. You've seen pyridoxamine before, it's drawn here. You don't have to draw it again if you don't want to, but in fact, I'll just copy and paste it, but I do want you to show me the alpha keto acid that gets formed. And I do want to see if you recognize that alpha keto acid, or I guess I should say alpha keto carboxylate. While you're doing that, I'm going to try to figure out this stupid eraser thing. All right, anybody got an answer for the ketamine? So it's pretty easy. You can take the aldimine. How do I need to change the structure of the aldimine to get the ketamine? What proton gets removed? Alpha proton. Good. Then where does it go? Onto the blue carbon. So what used to be my carbonyl carbon I'm frustrated enough, I'm wanting to like either scream or cry because the stupid eraser doesn't behave like it should anymore. And then I'm missing what? What am I missing between the alpha carbon and the nitrogen? The pi bond, right? So that's the switch. The carbonyl carbon used to be on the left, now it's on the right, and we switched that by tautomerization. Okay. Anybody feel like drawing the alpha keto carboxylate now? If you don't want to, you can volunteer as an unsuspecting victim. I'm just kidding. You two are pointing at each other, so one of you figure it out. Couple rounds of Rochambeau, we're tying the whole way. Okay. Dang it. So I want you to draw the product of, sorry, you got volunteered, I'm sorry, what was that? The hydrolyzed product,
Okay,
yeah.
okay.
Yeah, I want to see what the hydrolysis product of the ketamine is.
Okay. Oh, I don't have room.
You need space, so just circle that, drag some of that stuff out of the way. Yep.
I'll draw it down.
All right, great. That is correct. Very good. Now for bonus points, anybody recognize that? Ooh, good. Oxaloacetate. That's somebody we've encountered before, and that is an intermediate in the citric acid cycle. Now, that can be used for gluconeogenesis, but because this just takes the place of oxaloacetate in the citric acid cycle, it doesn't really get broken down or oxidized any more than that, because the citric acid cycle itself exists to oxidize acetyl-CoA. Okay, so if you were saying, well, how much ATP do you get from oxaloacetate? Well, by metabolizing it, well, none, because it just goes to become an intermediate in the citric acid cycle. Of course, if it became from, so this would be they call this well, there's a word for it that doesn't matter. It's a replenishing reaction. It replenishes an intermediate in the citric acid cycle. But your body can also use it for gluconeogenesis. Okay, your problem set is going to have you do exactly this thing, but with the amino acid glutamate, so that you can see what that goes to. But the idea is that by transamination, you're able to take alpha amino acids and turn them into alpha keto acids, which you can then either metabolize like pyruvate, or you can use further. In your problem set, you'll have to do something very similar, as I said, with glutamate. All right, yeah.
What happens with the other product?
Okay, yeah, great question. What happens with pyridoxamine, the amine? Yeah, because obviously you don't want that building up. You need to regenerate PLP. So the pyridoxamine, which is the product after hydrolysis of the ketamine, you're not going to like this, but what I've shown you is just 1/2 of what this enzyme does. It turns out that pyridoxamine is only a temporary carrier for the NH3 group. What will happen is that your body will then take pyridoxamine and it will take another alpha keto acid, in this case alpha ketoglutarate, which I'm going to draw for you here. This is also an intermediate in the citric acid cycle, 5-carbon alpha keto acid. it's going to do exactly the same thing that we did before, except in reverse. So you're going to first make a ketamine between the carbonyl carbon on the ketone of alpha ketoglutarate and the oxygen there, and the nitrogen of pyridoxamine is your amino group. You're going to tautomerize to the aldimine, and then you're going to hydrolyze the aldimine. And in the end, what you'll get is alpha ketoglutarate, except in the place of the carbonyl, you're going to have the amino group. Let me get the stereochemistry right. This is the amino acid glutamate, and that's going to restore over here the aldehyde on PLP. So PLP is a temporary carrier for amino groups, and it will deliver the amino group to glutamate, which is used for system-wide NH3 group transport. So when you go to ship a hazardous substance in the United States, you have to follow various regulations. This is why, for example, they don't want you taking certain lithium batteries on airplanes. This is why if you show up with a gasoline can in the airport, they're like, no. And if you want to ship gasoline, you have to have, like when we order chemicals, there are bottles packed inside other containers, which are themselves packed in other containers to prevent damage. Glutamate is like the super specialized shipping container for ammonia, because it can now deliver that NH3, which we took away from the amino acid somewhere else. And we won't talk about the fate of glutamate. Glutamate can get rid of that amino group in a similar way to how it got it, and there's some other ways that glutamate delivers ammonia for excretion to the urea cycle, which I said we wouldn't talk about. But for most aminotransferase reactions, if you want to write, if you go look up what the reaction is in biology and you want to see it summarized, you'll start, for example, with alanine, and then they will list alpha ketoglutarate as a substrate for the enzyme, and then the enzyme is called alanine aminotransferase, and the cofactor is PLP. And in the end, what you get is pyruvate, as we already showed, and then you get glutamate, whose structure I just drew above, and we'll just copy it and paste it there. So that's the full reaction from start to finish. And so you can see it's pretty complicated, right? You first take alanine, you make the aldimine with PLP, you tautomerize to the ketamine, then hydrolyze. That generates pyruvate and pyridoxamine, but you gotta have something to do with pyridoxamine, so that comes over to alpha ketoglutarate, makes the ketamine, tautomerized to the aldimine, hydrolyzed, that liberates PLP and gives you glutamate. And that's how amino groups are tossed and transferred around in biology. Okay, questions about that? Kind of overwhelming. Arrow pushing for this, if you can push arrows through the tautomerization step, you're good to go. Predicting the products, can you figure out where the nitrogen goes? Can you follow that through from starting materials to the aldimine, to the ketamine, and then to the new alpha keto acid and pyridoxamine? All right. So that's amino acid metabolism. Now, there's one more thing that I want to talk about that amino acids can do. Hopefully what I've shown you so far is that amino acid metabolism is intimately interconnected with carbohydrate metabolism. You've seen pyruvate, oxaloacetate, and alpha ketoglutarate involved in these things. What we'll do now, I wish I had time to tell you which amino acids were like glucogenic, meaning you could use them for for gluconeogenesis, versus which amino acids are ketogenic, meaning you can't make glucose from them. But because that has to do with ketone bodies, and I know that some people in nutrition and athletic training think a lot about ketone bodies, but we don't really have time for that. What I want to spend just the next couple of minutes doing is showing you how PLP is involved in a different reaction between amino acids to generate some well-known neurotransmitters and signaling molecules. So, for example, this is the amino acid histidine. So histidine's got this weird 5-membered ring side chain with 2 nitrogens. One of them has a hydrogen, the other has a lone pair. I'll use its 1-letter code. And then, of course, you've got PLP. And what we're going to do is do imine formation between the aldehyde of PLP and the nitrogen of histidine. So we'll draw the imine product. We would call it the histidine aldimine. Probably going to need a little bit more space. All right, let's draw the histidine portion with the histidine side chain. Here's the carboxylate. I'm going to draw, well, no, not going to draw the alpha proton. Here's the new imine double bond to the carbon of PLP. And then I am so sick of drawing the PLP structure. I am totally sick of it, as I'm sure you are. But we need it to explain what happens next. So here we go again. Okay. Now, if we were going to do transamination chemistry, we would tautomerize and our attention would be focused on the alpha proton. And remember, the reason for that is the presence of the PLP imine stabilizes a negative charge on the alpha carbon. Now, with that in mind, if the presence of the PLP imine stabilizes a negative charge on the alpha carbon, what else do you think could happen here? It's focused on the carboxylate. If the presence of the PLP imine stabilizes the negative charge, that means that all of this is a leaving group, which means that you can do a decarboxylation reaction. Kick electrons down, and then electrons go on to the alpha carbon as a leaving group. That's an irreversible decarboxylation reaction. You make CO2. What's left behind after that is what I'm drawing now. And this enzyme that does this is called histidine decarboxylase. There's a lot of enzymes that can do this for various amino acids. Oops, that's a negative charge there, a plus there. So here's the imine, and instead of, I'll draw the alpha proton there, instead of tautomerizing, we decarboxylated. There's the histidine side chain, and now the alpha carbon is negatively charged. So before, we removed the alpha proton to get a resonance stabilized, negatively charged intermediate. Here, we did decarboxylation to also get a negatively charged, stabilized intermediate. Notice that by resonance, I can put the lone pair / negative charge on each of those atoms. There may be a problem on your problem set that asks you to actually think about drawing resonance structures of this intermediate, resonance stabilization. If we protonate on the alpha carbon, that gives us a new aldimine that lacks the carboxylic acid. That should look like this. Man, if you weren't already sick of drawing PLP, I think you're there. In fact, you might be at the loathing stage. And if you want to sing the song from Wicked, you certainly can, about how much you loathe the structure of PLP. Unadulterated loathing. Okay, oops, and there's the imine. Okay, now we're going to do imine hydrolysis to get back PLP. Remember, in imine hydrolysis, we add H3O plus. We break the bond between the carbonyl carbon and the imine nitrogen. We put an oxygen attached to the carbonyl carbon that basically restores PLP and gives us this molecule, which is no longer histidine because it lacks the carboxylate group. What we've got now is histamine, which is a neurotransmitter and also is a signaling molecule for allergic responses. Okay? So this is how your body makes histamine. Now, what do antihistamines do? I don't think they inhibit this process. I think they inhibit the action of histamine. Maybe next time I will mention two other neurotransmitters, serotonin and dopamine, that also come from similar processes. But right now you could use a hit of dopamine and or oxytocin, and it's for sure you're not going to get it from me. So off you go. Have a good Thursday. I will see you tomorrow.